Tonight, we continue our journey through one of the most cutting-edge fields in science, the brain. Last month, we looked at how the brain develops during infancy and childhood. Tonight, we turn from the developing brain to the mature brain. Our subject, the aging brain. The study of aging is more important today than ever. Over the past century, life expectancy has skyrocketed thanks to a broad range of medical advances. The average American born today is expected to live for nearly 80 years. But as we extend the life of the body, we must also extend the life of the mind. With old age comes wisdom and maturity. However, there's also the deterioration of many cognitive abilities. The aging process affects memory in particular. Alzheimer's disease, dementia, and age-related memory loss are reaching epidemic levels. But cutting-edge research shows that these diseases may one day be preventable. Like every other brain function, memory is controlled by biological mechanisms that can be decoded by science. Over the past century, our understanding of memory has increased exponentially. We now know that memory comes in many different forms, each controlled by different brain systems. Aging degrades certain types of memory while leaving others intact. Joining me this evening is a remarkable group of scientists who have devoted their careers to understanding memory and the aging brain. They are Brenda Milner. She is one of the pioneers in this field. Her experiments in the 1950s formed the basis for the modern theory of memory. She is the Dorothy J. Killam Professor of Cognitive Neuroscience at the Montreal Neurological Institute and a professor in the Department of Neurology and Neurosurgery at McGill University. Larry Squire. His work has shown us that not all memories are the same. In the 1980s, he began categorizing memories according to their content, their purpose, and their locations in the brain. He is a professor at the University of California, San Diego School of Medicine and a scientist at the Veterans Affairs Medical College in San Diego. John Hardy. His research into the genetics of Alzheimer's disease has given hope to millions. In 1992, he discovered a genetic mutation responsible for the plaque that accumulates in Alzheimer's disease. He is a professor of neuroscience at University College London. Scott Small. He uses brain imaging techniques to study how the brain changes in old age. He is searching for behaviors, foods, and drugs that might prolong our mental acuity. He is an associate professor at Columbia University. And once again, my co-host is Dr. Eric Kandel. He is a Nobel laureate, a professor at Columbia University, a Howard Hughes medical investigator, and the man who is the inspiration, the guide, the spirit of this series. And it's always good to have him right here in this studio.
Pleasure to share this with you, Charlie.
What's important to understand about the aging brain?
There is good news about the aging brain. We are living longer, as you indicated, and we're living better because of public health measures, improved diet, increased exercise that people do, increased social involvement. People live better lives. The one thing that interferes in a number of instances with people living productive, mature lives as they age is an interference with memory. Memory is very susceptible to age. And therefore, one of the issues we want to address is what is the nature of memory? What is the biological base of memory? Where is memory localized in the brain? Now, as we discussed in earlier programs, we now know that most mental functions are localized to specific regions or combinations of regions in the brain. But the amazing thing is, until 1950, there was question whether memory storage could be localized to a particular region. Many people thought it could not. And then the two major heroes, the early heroes of the memory story emerged, Wilder Penfield and Brenda Milner. Wilder Penfield, an extraordinary neurosurgeon who trained with Harvey Cushing and also trained with Sherrington. Sherrington was one of the pioneers in brain research, and it really shows you continuities in scientific research. Great scientists don't simply do great science, they also train other great scientists. The specialty that Penfield developed was to operate on people with epilepsy. And he developed a particular procedure for doing that. He developed a procedure whereby he operated on people who were not anesthetized so they could talk to him. In fact, Sherrington wrote him at one point That's marvelous that you can operate on people's brains while they're talking to you. You have a preparation that speaks back to you. Working with monkeys and cats, I never had that. What he did is he infiltrated the scalp with a local anesthetic.
Yes.
He exposed the bone of the skull, and then he exposed the brain. And in order to make sure that he does not damage normal parts of the brain, he stimulated surface of the cortex. His specialty was epileptic surgery. And many of these came from scar tissue on different parts of the brain. So to make sure that it didn't damage, for example, areas involved in speech, which indicated in the orange circles on the right image, he stimulated different parts of the brain to see what the person's response was. We see here on the left, the right hemisphere, and we see here on the right, the left hemisphere. When he stimulated area of the touch area of the brain, patient felt touch sensation,
Right.
Not surprising. Stimulated the motor area, patients had movements of their hands. But when he stimulated the temporal lobe, which indicated here in these green markings, he found an amazing phenomenon, what he called experiential responses. The patient would all of a sudden say, you know, something is coming back to me, as if it's a memory. I hear sounds, songs, parts of symphonies. One person said, I hear the lullaby my mother used to sing to me. Amazing. Another person said, the segment of the symphony that I loved when I was young is coming back to me. So he realized he was getting close to something related to memory storage. And he called in Brenda Milner, an extraordinarily gifted young psychologist, and she worked with Penfield, and they showed that the temporal lobe, particularly its medial surface, is involved in memory. And they made several absolutely striking discoveries. They found that if you resected the temporal lobe on one side for surgery, you could stop the epilepsy, but you didn't interfere with memory at all. But if, for example, you removed one side, but the other side was damaged, so the person had an interference with both temporal lobes, person actually had a significant memory loss. They could no longer convert short-term memory into long-term memory. And then Brenda verified this. Penfield got a telephone call from a neurosurgeon in New Haven called William Scoville, who had operated on both sides of the temporal lobe. And he, in fact, had left the patient without any seizures. This is H.M., a very famous patient that Brenda studied extensively. But the person had enormous difficulty, Brenda found, in converting new short-term memory to new long-term memory. So he would meet Brenda repeatedly, but each time she walked into the room as if he met her for the first time. For the longest time, she thought this applied to all areas of knowledge, that the medial temporal lobes are required to convert short-term to long-term memory. And then she made another fantastic discovery. She found that in terms of a certain motor skill, H.M. could do it as well as you and me. So he retained certain memory capabilities perfectly well. Larry Squire entered the scene and began to explore this and found it wasn't just limited to motor skills. There were a whole family of perceptual and motor skills that people with medial temporal lobe lesions, bilateral, could perform perfectly well. And that made him realize there are two major memory systems in the brain. Those that involve the medial temporal lobe, he called this declarative memory, a memory for people, places, and objects. And there were a family of skills called non-declarative memory, motor and perceptual skills, hitting a tennis ball, golf swing, one does automatically. You and I are talking to each other. We're using the English language. It is presumably grammatically correct, but neither you or I are paying attention to my grammar. This is implicit. It's a remarkable insight. Once we knew about these two memory systems, the question arose, how do they age? How do they handle the aging process? Turns out the implicit memory system, the non-declarative system, handles aging quite well. But the declarative memory system, which involves the medial temporal lobe, is quite sensitive to the aging process. And it's susceptible to two kinds of disturbances: a gradual progression called age-related memory loss, and a very serious, rapid progression called Alzheimer's disease. We're going to discuss the differences between them, and we have here John Hardy, who discovered the first gene absolutely critical for Alzheimer's disease, and he showed exactly how it works. This is responsible, the beta amyloid gene, for terrible plaque deposits that is responsible first for killing synapses and then nerve cells. Moreover, now that we understand something about memory loss, we want to understand, how do we keep it? How do we keep intellectually engaged? And we realize that several things are very helpful: intellectual involvement, social involvement, and physical fitness. So we really want to understand is what allows Brenda Milner at age 91 to be the most intellectually capable person at this table.
All right, we're going to meet in just a second and pick up the conversation with Brenda Milner, remarkable woman at 91, and who is, as someone might say, as sharp as a tack.
Sharp as a tack.
So I have three questions. One, here's a book that you bring me every week, every month. You bring me a book, and this one is by Penfield, in which it's called The Excitable Cortex in Conscious Man, a series of lectures, I guess, that he gave. What's amazing to me is that we have in every instance, or in many instances, found out that these people on the frontier were amazingly prescient, and that later evidence, with all kinds of new techniques and new studies and imaging, showed how good they were, even though they were working a number of decades ago. It's amazing.
This is why they're called the giants of science. They not only have insight into what the immediate findings mean, but they can extrapolate them for their implications. And Sherrington was extraordinary for this, and he trained Penfield, who made a similar impact. We're still living in the Penfield-Milner era.
All right, this is really, really interesting stuff here. We begin with the remarkable Brenda Milner at our table for a conversation about these themes that we just were introduced to. Here is Brenda.
My task was to explore the functions of the human temporal lobe, about which really very little was known. And so there we see the left hemisphere of a typical brain, and we see the red areas of the speech areas in the left hemisphere, which you've already seen from Dr. Kandel. The typical temporal lobectomy, and this is the first important point that you do for the treatment of epilepsy, and is still a very successful way of treating epilepsy to this day, of this kind that has its origin in one temporal lobe, is unilateral. That's to say, the surgeon operates only on one half of the brain, and the other half is assumed to be functioning normally. We were able to compare the effects of removals in the left hemisphere, the speaking hemisphere, with those in removal on the other side. And there we got again an instance of localization of function within the domain of memory. There were small memory changes, and if it was from the left side, it was to do with memory for words, to language. If it was from the right side, it was affecting a little one's memory for faces and places and tunes, non-verbal, non-linguistic. But these changes were very mild effects. It was not anything of which the patients particularly complained, and often was present anyway before the surgery. And in exchange for that, the control of the epilepsy, when it was successful, as it usually was really changing their quality of life, and was also often showing improvement on general tests of intelligence. And all this was going very smoothly, and I was collecting all my results, my group data, when suddenly we had a really great surprise, and certainly for Dr. Penfield a great unpleasant shock, was when one patient, an engineer, a 46-year-old engineer whom we call PB, from New Jersey, came to have a left temporal removal. PB came back and had his operation in 2 stages because the first superficial one had not cured him. And after the second operation, this highly intelligent man, whom I had tested preoperatively extensively, said rather angrily, a little bitterly, "What have you people done to my memory?" And he had from then on what we call a continuous anterograde amnesia, a forgetting of the events of his life as he moved forward, so that he couldn't remember if his wife had been to see him that day. He couldn't remember what he had had for breakfast. He said as he woke up in the morning, it was buzzing, booming confusion. But then, in his case, gradually he would say that right now everything is clear. And all these patients, those few patients actually that I have seen with this kind of amnesia, this kind of forgetfulness, say this: right now everything is clear. So we have short-term memory preserved, but we have this inability to transfer information into a long-term storage. Now, we had this patient and wondered why we'd lost his memory, and were very worried. And a month later, we had a similar case. And at that point, Dr. Penfield was very worried, but also very excited and interested, as we both were. And so we reported these two findings in a meeting in Chicago. But we also had to come up with some explanation. And the explanation that we offered was that perhaps because we couldn't see into the brain beforehand, and because we never saw the other side, we only saw the left side, perhaps in these two patients there was some atrophy, there was some scarring, there was some damage in the medial temporal region, in the hippocampal region of the opposite hemisphere, on the right hemisphere, which was not operated on by Dr. Penfield.
This was the first demonstration that one could localize a memory function to a specific region of the brain.
Yes, yes. So. And so, so we reported these cases with this hypothesis. It was just conjecture for us in those days. And then Dr. Penfield got a telephone call from a neurosurgeon in Hartford, Connecticut, Dr. William Scoville, who called and said, "I've just read the abstract with interest because I think that the memory loss that you are describing in your two patients is similar to the memory loss that I have seen in a patient in whom I carried out my operation." Dr. Scoville's operations differed. First of all, he was operating on both sides of the brain. Our operations in Montreal were on one side with the hypothesis that was damage on the other. Here we're operating just on both sides. It differed also in not involving the lateral cortex, the lateral surface. And so here you can see a comparison between a brain of a control subject, a normal healthy brain, and the patient HM. This is a section through, and you're looking in the two hemispheres, and you can see that the medial structures, the deep midline structures in HM, which have been removed for a certain distance of about 5 centimeters going back. And now what about the patient? Because Dr. Scoville invited me to go down to Hartford, Connecticut, and study this patient and others. That's HM when he was young. And when I met him, he was young. I used to go around talking about this young man, but he died recently at the age of 82. So, but he was 29 when I met him in 1955. Now, HM also had seizures, epileptic seizures. And then at the age of 16, he began having really major convulsions, really very, very serious epilepsy. And it was treated with the maximum doses of the medications available at the day, and it didn't make any difference. He had great difficulty, although he was bright, finishing high school because of this. He had great difficulty keeping a job. He worked as a motor winder because he was having these big attacks and falling and so on. And during this period, Dr. Scoville suggested that perhaps this operation might help his epilepsy. He suggested that because he was impressed by the work that was coming out of Montreal from Dr. Penfield. Dr. Penfield's work was real pioneering, and Scoville was impressed that perhaps, even though this didn't look like temporal lobe epilepsy, perhaps these structures in the deep to the brain and the temporal lobes had an effect of at least encouraging and fostering epileptic seizures. So he proposed this, and he carried out this operation bilaterally on HM. And after that, it was immediately evident that he had this same inability to remember. It was described, he was in the hospital, that he could not learn the way to the bathroom, though he preserved all the amenities of ward life. That's to quote Dr. Scoville, that he was remained the polite, gentle, calm, pleasant young man that he was. But he and he knew Dr. Scoville because he'd known Dr. Scoville for many years, but that he did not remember the residents or the people that had been with him just around the time.
How many years did you work with him, Brenda?
Well, I met him in 1955, which was a year and a half after the surgery. And I used to go down to Hartford and test him, and we had him in Montreal for a week.
Did he remember you at any time?
Oh, no, never. He never, even when I would go down to Hartford and typically would spend 3 days. And during those 3 days, I could spend a session with him as we're talking across this table. I could be talking and working with him, and then I could go back. He would be in after lunch. I could meet him, and he would just look blankly. And I want to emphasize that he was such an extremely polite person.
How did you discover that there was any memory capability in him?
Well, the challenge for me was whether he could learn something. You know, this was obviously a challenge. You can't just say somebody can't learn until you've tried. And so I would go to over to the psychology department at McGill. I was in Montreal, and pick up 2 or 3 tests. I would choose 2 or 3 tests, take the night train to Hartford, spend 3 days working with HM, and I'd be putting him through his paces, going through all sorts of tests, various learning tests.
And what did you find?
And among those tests, I chose a motor learning task, which among them. And also something that always intrigued me about this is if somebody is practicing a lot or having lessons in tennis or golf or whatever, if you say to them, well, what did you learn today or this week? Of course, they can't tell you. Moreover, the attempt to tell you impairs the performance. So this is something that's going on. So there were reasons why I included a motor task in what I put down there, though I was, you know, I didn't know what I was going to find. And here you see a young man, not HM, practicing this task that I took, which is essentially that you're given a star, a shape of a star on a piece of paper with a double contour, and you're asked to start at the point of the star and trace a line, keeping within the narrow confines of the star till you come around to the top. That sounds easy until you realize that you have to do this only seeing your hand and the star as reflected in a mirror. And so at first we all do terribly because you get to the points of the star and you do this kind of thing. But with practice, we gradually learn. And the amazing thing was that HM learned. Now we see HM's performance on the first day. And what I am scoring here is the errors. The number of times he went out of the pathway and had to come back. That's an error. And you can see over those 10 trials that we did of the first day, his performance is getting better. Now, what would I expect? I'd probably expect that the next day he might be back at 0, doing as badly. And instead, look how much this has carried over, and he's continuing, not knowing, you know, that he's done this, but continuing. And then on day 3, look at that perfect performance. It's just amazing. If you look at day 1 and then
he does as well as Charlie Rose on this.
The thing that really impressed me at the end was he was a fairly tall man. This is one of my vivid memories, personal memories. He stood up like that, like this, and looked at what he'd done. And he said, he speaks rather slowly, he said, well, this is a little strange, he said. I thought that would be difficult, but it seems as though I've done it quite well. So this was an amazing dissociation between, you know, his total unawareness of all this experience he'd lived through these 3 days and the beautiful performance. And from this, you know, we speculated that maybe motor learning was mediated by some other systems in the brain. It was certainly not affected by the hippocampal medial temporal lobe lesion.
So this must have inspired you, Larry.
Well, it was fascinating because for a long, long time, we thought that motor skills were special and that everything else was memory and that everything else was what HM couldn't do. But eventually it was discovered that there's a whole family of abilities which are preserved in patients with damage in the medial temporal lobe. So it's not just motor skills, but also perceptual skills like learning to read upside down. Patients can learn to do that. And habits, things that you learn over a long period of time by trial and error, like learning to say please and thank you, or learning to wash your hands before dinner, Pavlovian conditioning, other simple forms of learning. So there ended up being a major distinction to be made between what we now call declarative memory on the one hand and non-declarative memory on the other hand. So declarative memory is what we mean when we use the term memory in everyday language. That's what's impaired in HM. That's what is dependent on the medial temporal lobe. And that refers to our conscious ability to remember the facts and events of passing days. But in contrast to that, we have non-declarative memory, which is a whole class of things that depend on different brain systems. The caudate nucleus, the amygdala, the cerebellum. So in the case of non-declarative memory, I mean, the reason it was so mysterious is that, as Brenda said, it is unconscious. It's a case where performance changes as a result of experience, and so in that sense it does deserve the term memory. Yeah. But in the case of non-declarative memory, we don't necessarily have any conscious memory content, nor do we even have the sense that we're using memory. So a nice example of this might be, say, the development of a phobia. Yeah. So a person at the age of 7 gets knocked down by a large dog, and then later on you could say, well, at least 2 different things have happened. On the one hand, the person may remember the incident, and that's declarative memory. It's a memory that can be brought to mind. It's conscious. They remember the fact and the event of the incident. But on the other hand, the person may be afraid of dogs. Well, that's a learned experience, a learned behavior.
An emotional
It's not even
experience.
experienced as a memory. It's experienced as an attitude or as a part of your personality. But it is a kind of memory, and it depends on the amygdala.
And how are both of these affected by age?
Well, they're rather differently affected by aging. Yeah, what declarative memory does weaken inexorably, really, by aging. This is a situation where 400 different people were tested at different decades on a simple list of 15 words and asked to recognize them later. And one can see a weakening across time, although one has to make the point that it's not enormous amount of memory loss. It's actually rather modest. And second of all, there's an enormous amount of variability in this process. We often like to say that as we age, we become more different from each other because we age in different levels of success. And it's even true in this case that 20% of the 70-year-olds are performing as well as the average 30-year-old. So there is an enormous amount of difference. So declarative memory declines like that due to changes that we understand take place in the hippocampus and the other structures in the medial temporal lobe. But implicit memory, non-declarative memory, tends to be more preserved, tends to be more stable. It's phylogenetically early, it's important, habits are important, but they don't change as much across time. One thing that's been very important in coming to all this is the importance of work with experimental animals in order to delineate all these systems. Indeed, even to figure out in the first place exactly which structures in HM were the ones that were important for understanding his memory deficit. Because that couldn't really be addressed until we had an animal model of the human condition in the non-human primate, initially in the monkey.
This finding that came out of the work of these 2 people really indicated that memory is not a unitary function of mind, that there are different kinds of memories, they're processed in different ways, and they're stored in different regions. A major, major advance.
And that's the essence of a summary of what we just heard.
That's right.
So pick up on that, Scott, if you will.
Well, what I've been focusing on is trying to understand the causes of this change in medial temporal lobe function, as Larry has already described. And as Eric already mentioned, there really are 2 major things one needs to consider as causes. One is normal aging itself. Just like aging affects every organ system, it also affects the brain. Within the brain, it's not a diffuse process. It seems to target select areas of the brain, aging does, and the medial temporal lobe is one of these areas. The other thing to consider, as Eric mentioned, is Alzheimer's itself, Alzheimer's disease. We know that Alzheimer's disease begins in the hippocampus, in the medial temporal lobe, presenting with mild forgetfulness before it sweeps throughout other areas of the brain, other cortical areas, causing a more profound cognitive impairment, something we would call dementia. And so it's sort of interesting. Here you have two processes, aging and Alzheimer's, these early stages of Alzheimer's, targeting the same general vicinity, the same general area in the brain. And so an important question is, can we try to dissociate those 2? And one way one might imagine doing that is based on the observation that the neurons in the medial temporal lobe, the brain cells, are not homogeneous. They're not all equivalent. You have different types of neurons that are organized into different parts of the medial temporal lobe. And it's because of that, because we assume that aging and Alzheimer's are mechanistically distinct, that they might target different parts of the medial temporal lobe. And so let me illustrate that. This is an MRI scan, magnetic resonance imaging. And as Brenda mentioned, the circle really is the medial temporal lobe. These are zoomed-in images of the medial temporal lobe. And these are fMRI, or functional magnetic resonance imaging scans. And they give you not only anatomical information, but also functional information. And so I've color-coded these images such that cooler colors reflect less function, whereas warmer colors, the reds, reflect more function. And I've also labeled two main areas in the medial temporal lobe that are most relevant. You have the entorhinal cortex, which is the main gateway into the hippocampus, and then you have the hippocampus itself. So on the left, you see the control subject. This is a healthy subject. On the right panel, this is a patient who has Alzheimer's disease. And you might appreciate that there's the most amount of blue or dysfunction in the entorhinal cortex, more so than other areas of the hippocampus, although over time, Alzheimer's does spread to include all of the hippocampus. Now, if you look at the middle panel, that's a subject who we think has normal age-related memory decline. And as you can appreciate, the entorhinal cortex is relatively preserved, the area that's affected by Alzheimer's. And instead, we see the most dysfunction within the hippocampus itself.
Talk a bit more about age-related in terms of how that works and what are we learning about that?
Well, I mean, I think perhaps the important thing to emphasize is that what used to be thought historically senility was the old term used for Alzheimer's dementia. And there was this assumption that senility had happened inevitably if all of us live long enough. But I think there's now a growing awareness that Alzheimer's disease is a disease, and by definition, a disease doesn't target everyone within a population. And so now you have these two processes or things that seem to be targeting the hippocampus, contributing to why, if we just sample a group of healthy 7-year-olds, many of them will have forgetfulness. Some of them have the earliest stages of Alzheimer's, and some we think have normal aging.
See, an interesting point that emerges from Scott's discussion is that until 50 years ago, this was a little bit of an academic issue because not that many people live to be 70, 80. Alzheimer's is now a disease we're much more aware of because many more people suffer from it because they live to be old enough to be susceptible to it.
Yeah, I mean, I think there are two parts to that. I think Alzheimer's clearly now is emerging as an epidemic, but so is normal aging.
Yes.
As Larry mentioned, it's relatively subtle, but as more and more of us are living longer and we all want to stay cognitively engaged in cognitively rich environments, even subtle forgetfulness is a problem.
Bothersome.
And I have many patients now who really aren't patients at all. They're just people who just notice the subtle changes. They're very healthy and vibrant. They're not patients at all. They're healthy, vibrant 70, 80-year-olds.
But they notice subtle changes in their ability of memory.
Now I think a really important point here is the limitations of studies in humans. And that, if I can illustrate, is one of the great utilities of turning to animal models. And what I've done here, I've oriented on the upper row is, again, the panel, the series of human brains you just saw. And on the lower row, I'm showing you the mouse hippocampus. As Larry already mentioned, there's a lot of it, a remarkable amount of similarities between the structures and the organization of the medial temporal lobes in a mouse and in a human. And again, I'm showing you now functional MRI or fMRI scans of mice down below. On the lower left panel, you can see a control mouse. And again, I'm highlighting the entorhinal cortex, the main gateway into the hippocampus. And if you look now on the panel on the right, lower right, that's a mouse in where we introduce a gene that causes Alzheimer's disease, and John will talk about this in a second. So these mice have been shown to develop Alzheimer-like changes at a relatively young age. And that's an advantage because we know with certainty who has the gene and who doesn't because we've introduced it. And we're not confounded by aging. We're not confused by the aging process because these mice are relatively young. And if you look at the panel on the right, I think you can appreciate that the dominant site of dysfunction in these Alzheimer mouse models is found, again, in the entorhinal cortex. Very similar to what's found in patients themselves, although over time it spreads to involve other areas. And now if you look at the middle panel, the middle lower panel, that's a mouse who we know with certainty has normal age-related memory decline. This is a wild-type mouse, as we call it. We don't introduce any genes, we just follow it across its lifespan, which is around 2 years. And we can image their hippocampus and medial temporal lobes. And again, as you can see in the middle lower panel, the entorhinal cortex is relatively preserved, and the sites that are targeted are within the hippocampus itself. So together we're starting to see this nice dissociation between Alzheimer's and aging, and perhaps to get to your question, we can use that information to get at underlying mechanism. In other words, in the mouse that has Alzheimer's, that gene is expressed in every part of the medial temporal lobe. In fact, in every part of the brain, yet there's one part of the brain, the entorhinal cortex, that seems to be differentially vulnerable or particularly vulnerable. Why is that? What is it about the entorhinal cortex that makes it vulnerable? If we can understand that question, we can perhaps get at mechanism.
Which brings us to understanding genes and what impact they have on these disorders.
Actually, my story, listening to Brenda, my story has a lot of similarities to Brenda.
Beautiful parallels.
You just hear it's exceptional patients and exceptional, people who give of themselves really and their families who make this research happen. Oh, yes. I mean, and that's what when I heard HM's story. So the first, for me, the story started with a family, which I'll show in the first image, a woman who wrote to me about her family. She's called Carol Jennings. She's still a friend of mine. She wrote to me about her family, and in her family there was really an exceptional type of Alzheimer's disease. Now, what we think of as Alzheimer's disease is late onset Alzheimer's disease, people in their 70s and 80s, but there are rare cases of Alzheimer's disease where it's very early onset. In Carol's family, the age of onset was about 55 years old, so multiple generations where the family had got Alzheimer's disease in their 50s. And actually, I know of other families subsequent to her family where people get sick in their mid-30s with Alzheimer's disease. The record is someone in their late 20s with familial Alzheimer's disease. And these families are really, of course it's terrible to be in these families in many ways, but they really offer us the opportunity to try and understand what causes the disease in those families. In Carol's family, what we can see is is that 3 generations affected by disease. We see that her great-grandfather had the disease, and her grandfather and great-uncle both had the disease.
So the red images show the disease and the yellow ones...
Squares are men, circles are women, and for obvious reasons, sort of thing. Red is affected, yellow is unaffected. And what you can see here is 3 generations. Carol is 1 generation below these 3 generations. So you see the great-grandfather had the disease, the great-grandmother was fine. They had 2 sons, both of whom had Alzheimer's disease, like I said, in the 50s. And then in the main branch of the family, you can see 5 out of 10 children had Alzheimer's disease, all at the same time. So all of the 5 siblings had the disease at the same time, and in the smaller branch of the family, the cousins, 1 cousin was sick out of 3. So we had 6 affected individuals and 7 unaffected individuals in that generation. And what we did was actually a conceptually very simple experiment. What we did was we looked at the inheritance of the genome in those individuals and asked the question, what part of the genome was inherited by all the affected family members, which their unaffected brothers and sisters did not? It's incredibly simple. It's not rocket science. It's really very straightforward. And what we were able to see is that the part of the smallest chromosome, chromosome 21, which is the diagram at the bottom, you can see that the 5 affected individuals and the affected cousin share a whole section of that chromosome. But you can also see that 2 of the unaffected individuals, the woman, the 9th woman on the left-hand branch of the family and the middle man on the right-hand branch of the family, have little parts of the chromosome, but they're not sick. So that tells us the disease gene is not in the bits that those unaffected siblings and cousins have. And it tells us the gene must be between those 2 bits. And when we looked carefully, we saw that the amyloid gene was in that bit, the bit that was only inherited by the affected individuals. Now, in fact, the amyloid gene had been identified about 4 years before, and it had been identified because the people interested in the pathology of the disease have been keen to find find out what goes wrong in the brain of people with Alzheimer's disease, what proteins are deposited, and they had shown that the amyloid protein was deposited in the plaques, and those horrible brown lumps there, they're about 1/10 of a millimeter across, and they're full of this amyloid peptide. So what our finding really showed was that in that family, in Carol's family, the disease started with that amyloid peptide. We found a mutation in the amyloid peptide gene which caused that protein to be deposited. So it was a very simple observation. It just tells us the disease starts with that peptide.
Fantastic observation.
But very
simple.
simple.
Gene identified that dramatically interfered with memory loss, the first gene identified in Alzheimer's disease, and opened up the whole study of the disease.
And so we and others have found lots of other mutations, but so that was certainly a start, and there was some great work from Peter Hislop's group in Toronto. He found that the families that we had not sorted out with amyloid mutations, in fact had mutations in a different gene called the presenilin gene, and we now know that the role of the presenilin gene is to digest the amyloid protein. So it just fitted together so beautifully and showed that all of these rare families have mutations in the same process, and this process was a process which led that peptide, that protein, to be deposited in the brain. So it was a great thing, and I captured from Brenda too the excitement you get. We all share this. It's why we're scientists.
Yes, it's very exciting.
You know, the excitement you get from knowing that something for the first time.
What is so interesting about this also is that it's only hippocampal medial temporal lobe memory that is lost. Implicit memory storage is for the longest time intact in people like this. And you can get people like de Kooning, the great artist, who with Alzheimer's disease could continue to paint rather interesting paintings because that to him had become an automatic task, like playing tennis is to you. Tell me about late onset Alzheimer's disease.
You know, that's a difficult, that, as you know, and that, you know, that's a difficult
That's why I asked you.
Yeah, that's a difficult. So we've been trying to work out what are the risk factors. There was some great work from Duke in the 90s that found the first risk factor for Alzheimer's disease. It's a gene called, for late onset, typical late onset disease, which is a gene called ApoE. Right. Great work, really a breakthrough. This is a gene involved in cholesterol metabolism. It was entirely unexpected. So that was great work. We actually don't really understand the connection between cholesterol metabolism and Alzheimer's disease, but that gene ApoE points to that connection. Then we've had 10 years of not really making much progress, but just this last 2 years, I'm proud to say back in England in particular, there's been breakthroughs using the new genetic technologies to try and find other genes. And what we're finding in these other genes is other genes involved in cholesterol metabolism, actually.
Also, I think it fits in with the general pattern that one of the predictors for good memory function is good health, physical fitness and good health, reasonable levels of cholesterol and things like that. Do you want to discuss this?
So there are a number of genes that are risk factor genes, and when we think about ways of modifying our risk, obviously currently we can't modify our genes. So the question is, are there other things that occur as we age perhaps that we can modify? And that perhaps gets to your question. And there have been a lot of interesting research into this area, but one story that's really emerging as quite elegant has to do with the way our body handles glucose as we age. So glucose, of course, is the main sugar, the main energy source as we eat a large meal. And the way the body handles glucose is it releases insulin from the pancreas, and that allows essentially the muscles to absorb the glucose. Now, it turns out as we age, all of us become a little bit insulin resistant. That's the term used, meaning the muscles are a little bit less sensitive to the insulin. And what that effectively means is that the pancreas tries to crank out a little bit more insulin, and glucose regulation is less stable. If that becomes profound enough, that becomes type 2 diabetes. That is a definition of type 2 diabetes. But even without that diagnosis, that's an age-related process. Now, it turns out that insulin and glucose are a two-pronged problem that are very relevant to what we're talking about. So a number of studies have shown that increases in insulin are a risk factor for Alzheimer's, whereas the changes in glucose regulation seem to target the areas of the hippocampus that are related more to normal aging and therefore are more linked to normal cognitive aging. So insulin and glucose, both part of the same metabolic pathway, seem to be players here. And perhaps the most important thing to emphasize is that there are ways in which we can modify those age-related changes. So a good example is physical exercise, aerobic exercise.
And what about social engagement? What about cognitive training?
Equally important. It's extremely important to be intellectually active and continue your career as long as you can possibly carry it on and do other things that give you intellectual pleasure. Linda Fried, who is the Dean of the School of Public Health at Columbia, is interested in cognitive function and aging, and she's found that if you take people in their 70s and put them into elementary schools as assistants, a volunteer corps, their intellectual function tends to persist on a fairly high level compared to controls that don't do this. So cognitive involvement, particularly in social situations, is very, very helpful. And certainly social involvement is extremely important.
Tell me how you explain your remarkable
Yes, Brenda.
thing. I mean, it really is.
Well, I'm going to have to put genes first.
Okay,
because
genes first.
Because
there you go, John.
She saw John here.
Because my mother, who had a harder life than I did, lived to be 95, and she worked. She was in music, different career altogether, and she worked teaching until the age of 88. She taught music and singing, and yes, till the age of 88, and she had to stop because she was getting deaf, which fortunately I have escaped so far, and so. Otherwise, she would have continued.
Okay,
and
so
she
genes is first.
Genes is first. Genes are first.
Yes, genes are first. Right.
British training.
Right. Thank you.
Well, I think engagement, I believe engagement in cognitive activities, but not solitary ones for me. It's very important to me to be engaged with young people. And as I say, when I was young, I liked to be engaged with older people across the spectrum of ages and on some problem that excites one's curiosity. I think my real secret is that I'm so curious. I'm extremely nosy about everything.
Oh, my God. That's the best news I've had all day. That's my profile. Right there.
That's your profession.
And the third thing is, although I don't take formal exercise of anything, I walk. I don't drive a car. I've never driven a car. I live near my place of work. I've been 60 years at the Neurological Institute. Right. Montreal is a hilly city, Mont-Royal. Yeah. And so I walk up the hill to work, and I walk back, and I walk all over that city. And I think that I do that because I enjoy walking.
What are you, 91?
Yes, 91.
91, okay. Where is the good news in all this in terms of memory?
So there are two parts to the good news, perhaps. One is that not everything worsens as we age. As you mentioned, there's wisdom.
Exactly right. That's what we haven't really explored that. There's something important happens that matches some of the loss of memory, which is a certain advantage.
Wisdom, perspective, that certainly matures.
Right.
Anxiety levels tend to decrease. So many aspects of cognitive function that don't require memory actually mature quite well.
It's also true that memory is built so that we, even as a young person, the way our memory system works is that we are always going for the gist. Right. We don't care so much about the details. I mean, thankfully, we don't remember every detail.
Good. Yes.
And so that makes it possible for abstraction and get the gist. And older people, then, that's something they can do.
Can I add to that two things? I mean, in terms of the good news, because there is some bad news. Let's be honest.
Yes, of course.
Particularly when dealing with Alzheimer's disease. So, the first thing is that the earliest stages of Alzheimer's does not immediately cause cell death and rampant cell loss. It's really characterized by loss of synapses, and I think in this program you've discussed the difference between right. And I think that's a very important point because as Eric and others have shown, there is plasticity. In other words, you can regrow synapses, but you can't regrow neurons. And I think that hidden in that observation is good news, because if we can detect Alzheimer's at the very earliest stage, before there's cell death, I think it's obvious to say that it's easier to cure or to fix a sick cell than a dead cell.
Right,
And so
right.
there's some good news in that. And I think the other...
Because for a long time, the notion was, and then I'm allowed to be the sort of layman here, for a long time the notion was that nerve cells died. And now we're looking at it and saying synapses between neurons
That's right.
is where the issue is.
First. Ultimately,
Right.
there is death, but that's where the functional imaging and various techniques to try to capture the disease as early as possible, that's going to be the window that's going to be most therapeutically efficacious.
It's not hidden away in the closet anymore.
That's a good point.
It's not hidden away in the closet. There's openness about it, and that of itself has brought. It's much better to be an Alzheimer patient today than it was 25 years ago. When I used to go around and see Alzheimer patients 25 years ago, they were hidden away in the back rooms of long-stay institutions. There's certainly been enormous progress. And
Nancy Reagan,
exactly,
made a real contribution because of Ronald Reagan and his decline.
Much better caring.
And Ronald Reagan deserves credit for that, too, in terms of announcing it early on.
Absolutely.
I think we want to remind ourselves, too, that we've learned so much about the cell biology and molecular biology of the pathways that lead to the deposition of amyloid, that every detail that's added to that knowledge gives us another potential drug target, gives us another potential way of intervention. That's understood a lot better.
It also points out, which many people don't appreciate, how difficult it is to get a really good drug for any disease, even once it's identified. Huntington's disease, we've known the gene for a long time. This is it's an essential beginning, but it doesn't guarantee immediate therapeutic efficacy. It takes a long time to develop a really useful drug.
This man has made some real contributions. He's too modest to talk about it in this whole area of understanding memory. Am I right, John, or not?
Absolutely right.
Sure.
This is a packed audience. These are my friends.
And what would that be? How would you define that?
Well, I mean, if we said that understanding the basic mechanism of an abnormality, Alzheimer's, you have to get to the basic cellular and molecular understanding. That's what the Kandelian project really began in the late 60s, or Kent Eric began. He really started dissecting out this complexity of memory down to the level of the molecules and the cells, and that's really been transformative.
So what's up next time?
Next time we're going to consider the emotional brain. Emotions are the instinctive forces that drive us to seek pleasure and to avoid pain. And this is such an important topic, we're going to have 2 consecutive programs on it. In the second program, we're going to consider misery, unhappiness, anxiety. In the coming program, the first program, we're going to consider how we seek pleasure and how addictive drugs co-opt the pleasure system for their own purposes. It's going to be a wonderful program.
I look forward to it. The emotional